Hello, and welcome to the science of Fox Chase Cancer Center. My name is Lauren O'Donnell. Most of us think about riding on the subway system as being a pretty routine thing. But scientists here at Fox Chase are using the idea of networks or hubs, just like a complicated system of subway systems and train stations, to fight cancer. Here at Fox Chase Cancer Center, the Golemis lab is using this concept of biological networks, which we call systems biology, to study cancer. The big questions they're asking are, why do some people get cancer? And why is it so hard to predict who will get cancer and who won't? And finally, why do some people respond so well to current cancer drugs while others do not? The answers to all of these questions may lie in the emerging field of systems biology. Erica Golemis, a scientist and professor at Fox Chase Cancer Center, explains.
We've gone from having no molecular description of what was going on in a cell to just a few genes, to tying the genes into networks, to now being able to take individual patients that walk in the door and generate full sequencing profiles of them and really very detailed molecular snapshots of their individual cancers. And so that is so utterly transformative in how we think about the disease. When I first started, people thought about, here is breast cancer, or here is, colon cancer, and now it's many, many different subsets, specific molecular lesions that are associated with each one, molecular drugs, tools that are available to try to target each one of them. So the types of questions that have become interesting are much more drifting to applied, how do we profile people more accurately? How do we use this knowledge to match individual drugs to individual patients in such a way that you can improve outcomes, reduce toxicity, maybe take drugs that are involved in targeting proteins in cancer and figure out whether they might be used as preventive agents or whether they might have uses in other diseases that have some properties of cancer. One of the advantages to working at Fox Chase is that we have absolutely outstanding MD-type physician scientists and clinical researchers who I absolutely love working with. So if you think of the work in this laboratory as focused on studying signaling mechanisms, on studying genes, on studying networks of how proteins interact with drugs in individual types of genetic backgrounds, then we take this sort of general body of knowledge and set of skills, and we're collaborating with clinicians who work on colorectal cancer, on lung cancer, on a little bit on pancreatic cancer. So we collaborate broadly across different disease types. One that we've spent a lot of time working with is head and neck cancer, which actually gets a little bit more personal because my father eventually developed head and neck cancer from having been a smoker in his early years, which he deeply regrets. He was treated here, he was cured. I became very interested in the disease. And we've been working for the past decade with clinical colleagues that are, specialists in this disease, which still clearly needs better treatments. We work with patient samples. We have something here called a biosample repository. This is patients very kindly donating samples from their tumors, or some cases their matched blood, so you can figure out their genetic background that isn't cancerous. And because this resource is there, it helps us parse out, specific mutations that are, linked to types of tumors that you might want to, treat in different ways. I'm still learning things to this day because we study things scientifically in the laboratory and they seem like good ideas. And then you start talking to the clinical people about, well, wouldn't this be a great idea for a trial? And they say, oh no, you can't combine this drug with this drug because of the side effect profile. Side effect profile is not something you normally think about if you're working with mice. Or you say, I'd like to take this molecule and bring it forward for pharmaceutical development. And someone with a chemistry background will say, no drug company is ever going to work on it because of, intellectual property, or because there's already something in the market that has a similar activity, so nobody's going. It's this very practical, or I should say pragmatic, concerns that sort of define the field where you can take your science forward. And because you have the opportunity of working with people who are, you know, in the front lines with this kind of issue every day, it sort of helps you identify what the better path forwards and where you should concentrate your effort. There's one project in the laboratory that I'm hoping we might have ready for, writing up for publication in the next year or so. And I mentioned earlier that polycystic kidney disease, in terms of signaling, has many features in common with cancer, even though the presentation is very different. In that, in the one case, you get a kidney that just fills up with cysts and ceases to function, and tumors, as most people know, that you get uncontrolled growth and they tend to metastasize around the body. That's something that polycystic kidney disease never does. And we think that there's an active role of the gene that's mutated there that is acting as a sort of a late-stage tumor suppressor that's preventing colon epithelial cells from actually invading and moving on in the disease. And so we've been crossing genetic models in mice for polycystic kidney disease onto colon cancer models and developing mice that we've been analyzing, and we're finding some extremely interesting results, and we're trying to understand how it's working. But it does look as if these two theoretically quite distinct diseases are sort of, because of this difference in property and invasion, it's telling us something fundamental about what causes a cancer cell to invade or not. So what inspired me to become a scientist? Starting as a very young person, I was always extremely interested in nature, in bugs and plants and animals of all different types. They're absolutely beautiful. They're fascinating watching how they behave. As I went on through high school and into college, biology classes were incredibly fascinating, and the people had a great deal of fun in them. Another thing was science fiction, classic science fiction from the 50s and 60s. Science was adventure, science was the ability to create new things and to, better humanity, and it was a very positive message. And the combination of the sense of discovery, the just fascination with living things, the fact that I had some inclination at that point that I might want to work in medicine, although I ended up doing research more than medicine. Biology was highly aligned with that, and it was just, so many things just push you together, and then once you start down that path, it just sort of pulls you in.
This concludes another edition of the Science of Fox Chase Cancer Center. For more information on the Golemis Lab or on other labs here at Fox Chase, please log on to www.fccc.edu. My name's Lauren O'Donnell, and thanks for watching.